going to be discussing about the advances and challenges in restoring vision for people with certain types of blindness, from both a neurobiology perspective and an engineering perspective. So before we get started, I just want to give a brief introduction of who I am and what I do, um, and then we can get right to business. So I I was born and brought up in Bangalore. My partner jokes about how my life revolved in that 5 kilometer circle in Rajajinagar eating CTR dosa. Um, so that, and then after that I went on to get my undergrad BTech in electrical and electronics engineering from NITK Surathkal. So when I was in my third year, I had the opportunity to do a research internship at the University of Toronto in Canada, where I was working on developing brain-computer interfaces with electroencephalography technology, to with basically imagined hand movements for people with paralytic stroke and motor disabilities. So this was sort of my first introduction to neuroscience from an engineering perspective. And I went on to pursue a master's at University of Southern California, where I specialized in signal processing, machine learning, and neuroscience. And then I worked at Intel on AI applications for a while. Now, currently, I'm a PhD student at the University of Washington, where I research about vision neuroscience. Specifically, my research interests include understanding the different emergent technologies for sight recovery technology to restore vision in people with blindness. So that's kind of a long spiel of what my journey. So now let's talk about vision. So before we talk about engineering aspects of sight recovery technology, I want to give a quick introduction to neurobiology 101 of the visual system. So try and understand what really it means to see, right? So, basically, vision is not just the eye, right? So it's a cascade of visual events that happen that starts from the front of the eye, but it goes all the way to the brain, which ultimately understands the light stimulus that we get and learns to make sense of it, right? So, as I said, it starts at the eye where there is a spot of light, like a source of light in your visual surroundings. And then that falls on your eye, and the lens at the front of the eye focuses these light rays and creates an image at the back of the eye called on the layer called retina. So this is classic 11th and 12th optic stuff, right? So now, an important thing to note about what the retina is that it's actually an extension of the brain, right? So, by that, what I mean is the cells of the retina are neurons, and they are the same cells that are present in our brain. It is just that it is specialized for vision. It tries to convert light signals into corresponding electrical signals and sets off a cascade of visual events, all the way to the back of the brain, right? So now, when you peer closely into the retina, you can see that it is comprised of five major layers, right? And the back of the eye that you see here is called the photoreceptor layer. Now, these are actually the sensors, and these are specialized neurons which can take the light signal of a certain wavelength, frequency, spatial frequency, temporal frequency, blah, blah, blah. And then that converts it to corresponding electrical signal, right? And then it sends it to the underlying layers. So these layers, and then this sort of gets bundled up. The innermost layer of the retina is called the retinal ganglion cell layer, and all of that gets bundled up into what is called the optic nerve, which goes, so this is like the top view of the visual system. So you have the eye, and then what you see here, uh, this is the optic nerve, and then it takes information to the back of the head. So the back of the head is called the primary visual cortex, and that is the sort of first line of defense, which where the brain starts to make sense of what we see, right? So why is all this important? Why am I showing you images of biology when you didn't want to study that? So there is an important part to it. I'll get to it soon. So let's talk about blindness for a moment, right? Blindness is not one specific disorder. It's more like a blanket term which is used to describe a whole bunch of disorders, right? And it can be categorized into different categories based on different types. Like for example, one of the categorizations is time of onset, right? So by time of onset, it means when people develop blindness. So in that, there are primarily two types. One is congenital blindness, where people develop blindness. They're born with it, like they are blind when they are born. And the second one is late onset of blindness, where people develop blindness with age, right? They were able to see at some point in their life, and then with age they slowly become blind, right? Now this is a very why is this important is that these are the problems with these are completely different, right? So there is a study of a person called Mike May. So he actually lost his vision when he was 3 years old, and he was blind for the good part of his life. For like about 40 years of his life he was blind. And then surgeons developed a surgical technique where they could bring back the sensation of light in his eyes. And then when they did the surgery, they saw that the eye was working fine. It was getting the input, and the retina was doing its thing. It was sending the inputs the right way to the brain. But he couldn't make sense of it. Like he couldn't, he saw everything as just blobs of light. Like, say, for example, you go to a movie and like there's like, you have a camera with like a water droplets on it, and then you just see everything as blobs of light, like bubbles, right? So that's kind of what he saw. He couldn't make sense of anything that he saw, which was very surprising because his eyes were sending information the same way as a normal person's eyes would send. But the catch here was that the problem was that when we start learning, like when we are 2 to 5 years old or 2 to 7 years old, our brain starts to develop the specialized circuitry to be able to make sense of what we are seeing, right? So those visual pathways have to get created from infancy, right? So he missed that critical period, and he was blind during that period, because of which there was no underlying circuitry which could make sense of what he was seeing, because of which he's functionally still blind. So the problems of congenital blindness are completely different. In late onset of blindness, where people had vision for a long time and then they developed blindness, the problems there are completely different. For them, they have the whole circuitry in place, right? Like the whole visual system would have developed over like their childhood. But there are different levels or in the visual system where they could have had an injury or some cells start dying off, because of which they develop blindness. So that's one categorization. Now, the second categorization is dependent on the root cause, right? So, what is it that's actually causing that blindness? So, one kind of categorization is maybe the blood vessels stop working, or they had some injury or accident, and so on. There are some external factors, right? One of the most common types of blindness caused due to vascular dysfunction, or like blood vessels stopping to work correctly, is diabetic retinopathy, right? And the second type here is actually a neurodegenerative cause, where due to some sort of inherited disorder, uh, people start they have genetic mutations, because of which their own immune cells in their body start killing off these retinal cells or brain cells, because of which the neurons or the retina starts degenerating and people start developing blindness, right? So that's one categorization. Now, the third is anatomical, like depending on which part of the visual system they have that problem in, right? The first is corneal, which is at the front of the eye, where there is some sort of a blockage that happens due to cataracts, maybe. Cataracts are the most common corneal forms of blindness, because of which it kind of blocks off, like there's a opaque filter in front, right? And the second is retinal. This is usually due to some retina damage or because of a neurodegenerative disorder, which is very common. And the third is some problem in the optic nerve. So in this diagram, like you can have issues here, or in the optic nerve, or cortical, like the whole circuitry, the whole system from the eye till the brain might be working just fine, but for some reason the brain's damaged, like they had an injury, or during fetal development, like when the baby was in the womb, they had like it could have had some sort of a neural development disorder and so on, which can cause blindness, right? So blindness is actually a whole blanket term, and what I work on, like my research interest, is specifically to late onset of blindness caused due to neurodegenerative disorders in the retina, if that makes sense. Basically, people who develop blindness over time. And this is actually one of the leading causes of blindness in our country, due to macular degeneration and so on. So that's a lot of biology. So let's get into the engineering aspect now. So the motivation for this is basically inherited retinal disorders are genetic mutations, as I told you, which lead to these photoreceptor cells dying off, which causes blindness in the retina, right? Some of the most common disorders are either retinitis pigmentosa, macular degeneration, and so on. And why does it matter? Because about 3 million Indians actually suffer from complete loss of vision, and about 21% of our population has visual impairment. And India is actually like the leading, is the country which has the highest number of people who are blind. So that's kind of the motivation for this research, right? So let's talk about retinitis pigmentosa for a minute. Let's try to get a visual of what this looks like. Like I'm a visual thinker, so when I see things, I will understand it better. So this is the cross section of the human eye, right? And then when you look at, there is something called the electroretinogram, which basically takes an image of the back of your eye. And when you see that image of a healthy retina, this is what it looks like. This is what it should look like, right? But in a diseased retina, in a severely diseased retina, this is kind of what it looks like. So all of the black spots that you see are the cells which have died off. And basically, this is a condition where people start developing blindness from the sides, and it slowly starts closing in. So you can't see anything on the sides, only centrally you can see for a small field of view, and then that starts closing in and people become completely blind, right? So this is what a diseased retina looks like. Now, there are many emerging technologies using different types of modalities which are trying to sort of cure this, or find a cure for it, right? Now, what is the catch here? As we saw in the retinal anatomy, uh, photoreceptor layer is just one layer in the retina, and then you have like multiple layers which get input from these photoreceptors, right? But because the photoreceptors are not working, although the entire underlying circuitry exists, it does not get any input, because of which they cannot see, right? So what these technologies are trying to do is they're trying to find ways to bypass the photoreceptor layer, and try to elicit cell responsiveness directly or send input directly to retinal ganglion cells, right? And one of the promising technologies which has received a lot of attention in the past are electrical implants. So here, basically, what they do is they put an a bunch of electrodes, an array of electrodes, through surgery. They put it at the back of the eye, and then they use electrical implants to convert this light signal to corresponding electrical signal to activate these retinal ganglion cells. So it's kind of like bypassing the photoreceptor layer. And I'll get into the details of that in a bit. And then the second technology is called optogenetics. So this is of my primary interest, and this is where most of my research efforts are spent. I'll get to this in a bit. Let's talk about electrical implants. Um, Before I go on to the next slide, there are explicit surgical images coming up. So if any of you feel nauseous or queasy looking at like blood and eye and everything, just close your eyes or turn away for a bit. Okay, let's talk about what electrical implants, retinal implants do, right? So the basic idea is this. You have sort of sunglasses which have camera mounted on top of it. So this reads the light input from the surroundings. And then that input goes to something called the video processing unit, or VPU, which has, it's like a tiny computer which goes on the belt of these patients. And what happens here is it takes the light input and it has a model built into it to convert the visual input to corresponding photoreceptor electrical output, right? It encodes that. It has a neural model which encodes that. And then that signal goes to an array of electrode which is mounted at the back of the eye, like surgically in the retina. And the corresponding electrodes start activating retinal ganglion cells, thereby eliciting cell responsiveness, right? So this technology actually got a lot of traction in 2010-12ish, where Argus 2 was the first retinal implant device, which was approved by the FDA for people with severe retinitis pigmentosa. So about 200 people got this surgery done, and they signed up for this, and everything, it kicked off. So it includes a camera, VPU, and then there is electrical stimulation, right? So just to give you an idea of what it looks like, I'm going to show you the image. This is basically, they start the surgery at the back of the eye, and then this is the electrode that is implanted. It sits on top of the retina, and this is kind of what it looks like. This is basically like an array of electrodes, and then it sits on top. This is the ERG image with the retina, right? So this is a tiny electrode, like it's about 124 by 124 electrode, right? Now they've updated it to 256 by 256, but whatever. It's a very tiny electrode which covers a very tiny space. And just the catch, or like the USP here, is that you need to know how to encode the neural code. Like you need to know how this visual information translates to electrical signal. So that's the catch here, right? So okay, this sounds like science fiction. At least it did to me when I learned about it for the first time. But what is the issue with this? There are like a myriad of problems that come with it. First of all, you need to know the encoding problem. Like you need to be able to develop a model which accurately converts the light input into photoreceptor output, as I told you earlier. So assuming that you did it perfectly, like you came up with an amazing model which will exactly replicate what the photoreceptor is doing, there is a second problem that comes with it. Basically, what this is assuming is that the retina, the photoreceptor cells are basically all one type. Like it doesn't distinguish between different types of cells. But in reality, that's not the case. Many of these cells have different subtypes, and they are encoding for different things. Like there is a group of cells which are encoding for contrast information in the image. If you guys are familiar with image processing and so on, you would know that there is an edge detector, there is a contrast detector, and there is a color detector, and there is like a frequency detector, and so on, motion detector, right? So all of these different types of cells are trying to encode different things, which are not taken into consideration here. So that because you are sending the same input, assuming that it's all one type of cell to retinal ganglion cells, there is perceptually there are distortions. Like you won't people with this won't be seeing what we would be seeing, right? And the third thing is spatial distortion. So depending, our brain doesn't work like a camera, in the sense that it does not take pixel by pixel information and try to translate it in the brain. That's not how, no, that's not how it works. Basically, there is a model where the central one part, like if you hold out your stick out your thumb, just that just the region which covers your nail, like the thumbnail, that is the central vision, and that has 30% of your visual cortex dedicated to it. And then as you slowly move away from the center, the resolution, spatial resolution, becomes smaller and smaller, right? So depending on where you put this array of electrode in the retina, it can lead to different kind of distortions. It can make it blurry, it can make the percept look more, or it can detect motion with lesser frequency, and so on, right? And the third thing is temporal distortions, right? So basically, when there's something moving very swiftly, or like there's a bus which is going past you, you are able to, it is say moving at 30 kilometer per hour, right? And you're able to detect it, but that is only because your brain is sensitive to those temporal frequencies. If your eye was not sensitive, you would, it would be invisible to you, right? So because the speed with which this translates the information might not be the same as the speed with which photoreceptors translate information, this can lead to some temporal distortions, right? So just to give you an idea, this has been the crux of my professor's work, where they developed, and to understand all of this, you kind of need to quantify it. Like this is all very up in the air. You don't have a measure for this, right? So this is the crux of our lab's work, where we are trying to sort of develop quantitative framework to measure all of these systematically and kind of understand how to improve this technology. So it's imagine like in the 1900s when people were developing aeroplanes, right? Like they probably did not put people on prototype aeroplanes and get them to fly off. Like they would, crash and burn probably. So what was important was to develop a simulation framework which would kind of simulate all of the aerodynamics and know when the flight is going to work, when the flight is going to crash, and so on, right? So that's kind of what we do for site recovery. We try to quantify all of this and say what kind of stimuli don't work, or when, or what are the conditions in which this site recovery, this electrode will work and this won't work, and so on. So we have different perceptual models, as we call it, where we are trying to predict what people with these therapies would see, right? So the image that you see on the top left here, it's a Snellen eye chart. This is very common for any of you who've been to an eye doctor. You're familiar with this, right? And those of you who are not familiar with this, you're the lucky ones. So yeah, basically this is a measure of our visual acuity. So when you show this to a person with these electrodes, this is sort of what they will see, because the retina doesn't encode for pixel by pixel information. It's all like streaks, right? So, and this is the best case scenario. And just applying the same model to any random naturalistic input, this is what the output would look like in the best case scenario. So from this you can, and there are like additional distortions that can occur. So this is an illustration of what it would look like as you put the electrode away from the central vision or central visual field, right? So imagine this is the Snellen eye chart. Instead of it being projected at the center of the eye, it gets projected farther and farther out. So this is what it would look like. So lambda equal to zero point 0.5 means it's in the center, and lambda, as lambda increases, it goes towards the peripheral view, right? So as you go towards the periphery, or the electrode is placed in the periphery, it starts getting more and more distorted. So these models kind of suggest that while electrical implants have great potential to somewhat bring some sort of vision to people, it's definitely never going to be the same as naturalistic vision. But you want it to be functional, like you want to help people navigate like a visually sighted person. Otherwise, there's no point to it, right? So that's it, this is all in that direction. And then Argus 2 had a lot of attraction, and we learned a lot from it. However, there are certain issues with adaptation, and with the electrode sort of coming away from the retina, detaching from the retina, and so on, because of which this company is not operational. But there is a second generation of electrical implant companies which are trying to understand the predictive model before they go and put it in actual people. However, electrical implants do have one advantage in that they can encode time very well. Like it's very fast, so it won't sort of make fast moving objects invisible. But that's kind of the story of electrical implants. So I know it's a lot of information packed in, but are you guys good with this? If you are, then I can go to the next part. Um, just give me a thumbs up or something. Okay. Um, that's the story of electrical implants. Now there is another major player in the field, and it's called optogenetics. So probably I think 20, 25 years ago, some marine biologists were studying algae in the sea, and they found that there were certain algae which actually lit up when sunlight fell on them, which was crazy to think about it. And then when further studies were done, they realized that there are these proteins, specific proteins called optogenetic proteins, which are sensitive to light, and they essentially act like transducers, which convert light to electricity, right? And this is crazy. And when people try to study the chemical properties of these proteins, they realized that the photoreceptors also have similar proteins in their eyes. So now a whole research field emerged out of it, where people are now trying to take these optogenetic proteins and package them genetically, package them in something called AAV vectors, which are basically carriers. And then when they inject these proteins into the eye, these proteins go and bind to the retinal ganglion cells, making them light sensitive. So they're kind of converting these originally non-light sensitive cells to become light sensitive, thereby trying to elicit cell responsiveness in these retinal ganglion cells. Now this is crazy. This is also science fiction, and the great thing about this is you don't need surgery, you don't need complex setup where you're putting implants in the eye and so on, right? So this was a game changer. But let's talk about the problems. Like with it come a lot of different myriad of research opportunities to go and study it more systematically. So essentially, how the brain works, or right from the retina, how it works or how it transmits information, is that it sort of acts like 0 or 1. Like it either is in rest state, or when it tries to transmit some information, it transmits an electrical impulse. So it's a 0 or 1 sort of a code that it transmits into. And the information is encoded in the time duration between each of these electrical impulses that arise, right? So this is characterized by the frequency or firing rate of a particular neuron, right? And when the firing rate is high, you know that, or when there is an input and this neuron goes whoop, and it just keeps firing electrical impulses, you know that it is sensitive to that particular input, or it is specialized to detect that input. So that's kind of how the retina, all the brain, from the retina all the way to the rest of the brain, transmits information, right? So now people took these optogenetic proteins and they studied how retinal ganglion cell responses looked like in a normal retina, where they were getting their inputs from photoreceptors, versus in a diseased retina where these optogenetic proteins were sending input to the retinal ganglion cells to respond, right? So this is sort of what they found. So what you see on the left is one retinal ganglion cell response to a visual stimulus in a healthy retina, where these retinal ganglion cells are getting input from photoreceptors, healthy photoreceptors. And what you see on the right is the response to the same visual stimulus in a diseased retina, but the photoreceptors are all fried, they're gone. And the RGCs are responding based on the optogenetic proteins, activation, right? So clearly it looks very different. And if each impulse looks this different, that will clearly lead to certain perceptual distortions. So that is where we come in, and we started to develop something called the virtual patient framework, where we wanted to try and predict or come up with a model to predict what the output would look like, or what these people with optogenetic therapies might see, right? So for that, we started to develop a model, a mathematical model, to predict the neural response of these optogenetic proteins for different visual stimuli of inputs of different spatial and temporal frequencies. And from that, we use that simulated optogenetics to measure visual acuity, like the Snellen eye chart, and contrast sensitivity, and so on. These are like established metrics for to measure how good your sight is, right? So I'll come to the second part in a bit. I'll first talk about the model of mathematical model of the neural responses. So in order to do this, we use two state-of-the-art options which are being developed. One is called CHR Mine. Essentially, it's called the photocurrent model. And I don't want to go into the mathematical details of this right now, but what we intuitively, I want you to try and understand what this is doing, right? Essentially, imagine it to have four states, like closed state and open state. And each of the, like when you get a photon, which is incident on this option, it tries to convert it from closed state to open state and then back. But the speed with which it does it, and the level to which it does it, vary from one option to the other. So the speed characterizes the kinetics, or how quickly the option responds to light. Like, say for example, it's a very slow option, like in this case, like the reacher option. You have one image, or one photon, or one source of light which is incident on the eye. It will take almost 500 milliseconds to send a response. Whereas in a natural photoreceptor, it takes 2 to 3 milliseconds, right? Which means fast moving objects will become invisible to you because it's not keeping up with the movement of the visual stimulus, if that makes sense. Now, the second aspect is the sensitivity, right? As in, what is the amount of current that it generates when light is incident on it? Now, CHR2 is the OG option, which was found in these algae, but it only elicited current in picoamperes, and our eyes almost do it in tens of nanoamperes, which means that you need to blast the eye with a very, very, very bright light for somebody to see, for somebody with this optogenetic therapy to see what you're seeing in a low light condition. Does that make sense? So that characterizes the sensitivity of the optogenetic protein. We used this photocurrent model to be able to predict the responses of different opsins, and this is kind of what it looks like. The wider this curve, the slower the opsin is. So this from this graph, which opsin do you think is the fastest? Anyone? I I don't know if it's easier to interact. Okay, so I'll give you the answer. Basically, if it is not wide, the narrower the curve, the faster the option is. So this CHR2 is the fastest. But when you look at the responses, this has the highest peak current, which means it's most sensitive, right? So we took this CHR mine option and modeled the output, or predicted the neural response of this option. Um, Now, let's see what that looks like when you pass a specific type of visual stimulus. So, what we are seeing here is basically input square pulses in red. That's the visual input, and the blue curves are the visual output for these different options, right? So, what you see is that um, when it's one, the the visual stimulus is one, it means the light is on. And when it is zero, it is turned off, it's like completely dark. And we are trying to measure what the current response looks like for two different options. One is called the 4x bgag, blah blah blah, it's too long to pronounce, but that's one option, and chr mine is the other. So what can you observe here, right? The graphs on the left indicate the output for one option for these square wave currents, and the one on the right indicate the output for the second option, sorry, chr mine, right? So when you observe the outputs of the option on the left, you can clearly see that as the frequency of the visual stimulus increases, the response goes down, right? Like the peak response goes down, the amplitude is lesser, right? And it also starts getting distorted. It looks more and more like a triangular wave, and then it just disappears, which means but it starts off at the same peak frequency or peak output as this, right? So what I'm trying to show here, whereas on the right side, the chr mine option on the right side, it kind of keeps up as you increase the frequency. Even at 30 hertz, it is moving up and down similarly, right? So however, the peak response is lesser than the one on the left, which means that the option on the left has higher sensitivity but cannot keep up with faster moving objects. But the option on the right, on the other hand, is the opposite. It has lesser sensitivity, but it can keep up with fast moving objects. In other words, the option on the left, basically you can see things better, or you can start seeing things, you won't be blind with like regular lighting condition. But with the option on the right, you need to bleach the cones, like you need to have floodlights to be able to see the same thing. That's kind of what it says about sensitivity. About temporal kinetics, it's the opposite. The one on the left basically cannot keep up with speed. So if you have, it can maybe help you read a book, but if there's a bus which is moving past you, it will be invisible. That's kind of what it says, right? So different options have different advantages, and we need to. The idea of this project is to be able to understand systematically what these different parameters are, which will sort of uh, come together to create a visual perception for us, and how we can make it as functional as possible. Does that make sense? Okay. So this is the same thing, but with sinusoidal inputs. Um, and then let me show you. This is a fun movie. This is basically passing a movie or a video and through the model to see what the optogenetic distortion looks like. And these outputs are for the opsin which has highest sensitivity. So just to give you an idea of perceptually what it looks like. So imagine this is like a donut or like ring which is moving past the screen, and this is what you as healthy individuals with like normally sighted people would see. However, people with optogenetic vision might see it like this, right? That blurry line which is going behind it is because it's unable to keep up with the speed. And the faster it becomes, it disappears. So that's kind of the idea. I think I have one more. Yes, I have one more illustration just to kind of drive home the point. So assume this is like a naturalistic scene. You're sitting, you went somewhere. You're sitting in a cafe, just going about your business. So this is what a neurotypical person would see. Now this is what the model predicts it to see. Now, I would like to draw your attention to this lady or this guy moving, like swinging their arm and moving. Observe how their hand disappears as they keep swinging. Like it just disappears. It's not perceptible anymore, and that is not a glitch in the model. That is what's happening. Like people, fast moving objects will disappear. So that is what this model is saying, like it has to be fast enough. Now, how do we? Okay, now we have a prediction of the neural response of these optogenetic proteins. So, how do we take that and actually systematically measure and say, oh, you have 20 by 20 vision, or you have zero point 0.5 diopter issue and you need glasses or something like that, right? So, in order to be able to measure the visual acuity, we use something called psychophysics experiments to be able to measure it. So a little bit of stimulus about stimuli and perception. So I'm not sure how many of you are familiar with Fourier transforms, but if for those of you who understand signal processing, this is kind of a reminder or refresher for those of you who are new to it. This is like a crazy concept, and you should, as every engineer, you should learn about Fourier transforms. It's fun to know about it. So anyway, essentially what it says is whenever you have any image, basically you can represent it as a linear combination of sinusoidal gratings of varying spatial frequencies, right? So when you take a Fourier transform of this, you can kind of break it down into a combination of different sinusoidal gratings. And then so if you know the output of this of any system, right? If you know what a system does to all these sinusoidal gratings, you can put them all together and predict what the output would be for any generic image, if that makes sense. So you don't have to go about curating like thousands and thousands of different image data sets and keep showing one by one to measure or see how they would perceive it, right? Or what the eye does to that image. You can measure uh, an array of spatial frequencies, sinusoidal gratings varying in spatial frequency, and then predict the output of that. So that's about the stimulus. Now, our retina kind of acts like a spatiotemporal filter, right? So, by that, what I mean is our sensitivity is not the same to all frequencies, right? So, what this shows is that for sinusoidal gratings of very low frequencies, our sensitivity is actually lesser, which means that we need to bump up the contrast in order to be able to see it. And as you increase the spatial frequency somewhere in the mid region, our sensitivity is very good. So even with a very low contrast, we can detect those sinusoidal gratings. And then as you slowly increase the spatial frequency higher and higher, our sensitivity drops. So the red curve that you see here is the sensitivity curve, right? So it starts off low, it goes up to mid frequencies, mid spatial frequencies, and then it falls down. Right? So this is the visible range, and anything beyond that is invisible to us. Like we cannot perceive it, right? Um, and this is in spatial domain. The same thing happens in the temporal domain. Now, there is a metric called spatiotemporal sensitivity function, which basically tells you or which it's basically a mathematical function, which is a function of both spatial and temporal frequencies, and sensitivity plotted as the dependent variable. So that if you have that function, you can kind of tell at what frequencies people see it, like what frequencies are visible to people and what frequencies are invisible to people. Now, this spatiotemporal sensitivity function remains almost like more or less the same for people with normal eyesight or neurotypical vision, right? But with optogenetics, this might be a good measure to see if that curve, like the optogenetic curve, matches the neurotypical curve, right? So that's essentially what we wanted to measure. And in order to measure that, we designed an experiment, a psychophysics experiment, to go about it. So what we did was basically we showed this is a very standard paradigm to measure it. You design an experiment where stimuli are forced to choice, which means you get one of two inputs, and people have to detect whether they can see it. Like they have to respond saying whether they saw a grating which was oriented to the left side or something oriented to the right side. And what we change is we keep changing the spatial frequency, and then we also change the temporal frequency, like how fast or slow it changes, both in the sinusoid as a sinusoidal function. And then we also change the contrast. So we make it dimmer and dimmer, like contrast reduces and then contrast increases. And we want to find that sweet spot where if we reduce the contrast at this temporal spatial frequency and temporal frequency any further, it becomes invisible to us. If we increase it above that, they start seeing. That's the idea, right? So this was the experimental paradigm. Basically, we presented the grating, which was oriented in one of two directions, and participants were asked to identify the orientation by pressing either the right or left arrow. And temporal frequencies, we varied between 1 to 18 hertz. This is like the range of human perception, or like most sensitive range of human perception. And then spatial frequencies, we varied between zero point 0.5 to 16 seconds per degree. And contrast levels were modified based on a Bayesian adaptive procedure that we used. Right. And then we collected different trials for like in baseline condition, where basically we showed these gratings to people with normal eyesight, and then uh, got the spatiotemporal sensitivity function for that. And to measure the optogenetic mediated spatiotemporal sensitivity function, we passed the same input through our model of simulated optogenetics. So essentially, the natural scene that you saw, systematically to measure what that would feel like, we pass it through these spatial gratings and then spatiotemporal gratings, and then we measure the temporal contrast sensitivity function. So that was the experimental. And then, yeah, so that's essentially what I'm talking about. There were three conditions. One was for normal eyesight, like neurotypical, just regular grading. Second was with opsin 1, and third was with opsin 2. Like we changed the models to simulate two different opsins. And this is still ongoing research work, and these are the preliminary results that we are seeing. So what you see here is basically contrast sensitivity function curves for three different conditions. The green corresponds to control condition. Basically, that is what a person with normal eyesight or vision would have. Like that's the sensitivity they would have. And this is log sensitivity. So this is in log scale. So every 1 point means 10 times reduction in sensitivity, right? And what you see here, the blue curve corresponds to the first opsin, the one on the left that you saw, the 4xb gag opsin. So, if you recall correctly, what we observed there was that the sensitivity was pretty good at lower temporal frequencies. But as the frequency or the input became faster and faster, the sensitivity dropped much more quickly. And that's exactly what we're observing here, right? So, the fact that it is matching at the lower temporal frequency means that you can read a book similar to a neurotypical patient. Not exactly, but close. But if there is a moving bus, they can't see it. That's the idea. And the red curve that you see here, that corresponds to the second opsin. And what this is trying to say is that it keeps up with our opsin, right? It keeps up with the visual stimulus because it's pretty fast, but the overall sensitivity is less, so the whole curve is kind of pushed down, which means you need to bump up the visual input light, like the surrounding light, or the in order for them to see something. Like, say for example, they have a book, what you could read in like I don't know, like 20-watt bulb, they would need 200-watt bulb to read it. That's the idea, right? So that's kind of where we are in our research. And okay, that's essentially what I have in my discussion. So there are different parameters. What we're trying to establish is that there are different parameters which all determine what vision looks like. Like, all of these parameters have to come together for us to see what, or make sense of what we're seeing, right? And these different options have different parameters, which are like different knobs that you can tune up and down to, and you can kind of create different percepts for the same input, right? um, and as we observed, opsin chr mine has temporal kinetics but lower sensitivity, and the other opsin has higher sensitivity, lower temporal kinetics. So the applicability of this framework is like the flight prototyping scenario that I was talking about. It basically helps you predict what people would see before you go and put it in them. Like, one of the biggest challenges with anything related to biomedical engineering or health sciences is that it's a chicken and egg problem, right? You don't unless you have something working really well, you cannot go and put it in real people. But unless you have data or you have tested it on some people, you won't know exactly how it works. So it's kind of a chicken and egg problem, and that's why research in biomedical sciences is so difficult. But the idea is you kind of find workarounds. You try to find ways creatively to be able to simulate and predict what it looks like, or what to understand whether your engineering device or implant is doing what you're expecting it to do before you go and put it in people. So that's the applicability of this framework. So you can systematically quantify the performance of these different options or even electrical implants to tell what it looks like, so that people who are designing it can go and make modifications accordingly. And yeah, we covered a lot in this, but just another note or comparison between these technologies. Like, you would never know what kind of technology is most applicable for a certain patient. Say you're a doctor sitting in your clinic, and somebody comes and they're like, 20 years down the line, they're like, there are so many different options that are available. Which one do you think I should get? Do I get an electrical implant? Do I get an optogenetic protein? Blah blah blah, right? So the idea is that electrical implants are still very good with temporal resolution. They're very fast, so you won't miss a moving bus. But the problem is the field of view is so small. There is so many distortion, blurriness, and so on, that in the state that it is in right now, it is not functional. But it gives better advantage in terms of temporal resolution. What optogenetics has to offer is it has great spatial resolution, because these proteins can, like, use these viral vectors to go and bind to these RGCs, so it can cover a wider range or wider field of view. But where it lacks is that it's slower. The kinetics of these proteins are slower. Unless you improve that, that won't be functional, right? So and one other big advantage of this is that it is less invasive than electrical implants, because you kind of inject it into the eye. It's still painful, still minimally invasive though. So and again, depending on the kind of blindness, you let's say these are applicable for people who have retinal degeneration, right? But what about people who have blindness because there is something, a damage in the brain or in the optic nerve, like diabetic retinopathy and so on, right? No, sorry, glaucoma, my bad. So in those cases, there are electrical implants now being researched, which can be directly implanted in the brain and not in the retina. So there are a lot of different technologies which are emerging in sight recovery. And there's a lot of research potential to go and study these things. And the idea is to try and make it as systematic as possible, so that you prioritize the safety of patients first, but also develop these these implants or sight recovery technologies, to be able to help people with blindness as much as possible. So that's the crux of my talk. Oh, before I finish, I have some useful info, which I was thinking that it would be helpful if somebody told me when I was in my undergrad. So a lot of these there are multiple opportunities if you're interested in research to go and do these things, but a lot of times we won't know where to start and who to ask. So just a list of some funded scholarships that are available for undergraduate students in India. There is one thing called Indian Academy of Sciences Summer Research Fellowship Program. So this basically selects certain undergraduate students to go and do research internships in popular research institutes all across India. And this is targeted for undergraduate students in their second and third year. I think they have fellowships after that as well, but I'm not 100% sure. The second is Raman Research Institute Visiting Student Program. And I was a beneficiary of this when I was in, like, I finished my second year. Basically, this is again a research internship where you can go and do one research internship in Raman Research Institute in Bangalore. And they have astrophysics research, they have brain computer interface research. It's quite diverse, and it's a very good insight into research world. The second thing is it's the USIEF Fulbright Nehru Scholarship Program, and this is a funded program. I don't think this is to do research internship. It's more like they give you the money and you use it for your own research with a professor in your institute or elsewhere. And this has different categories all the way from undergrad to postdoc level. Um, these are some of the ones that come to my mind. And there are also funded scholarships for undergrad students to do research abroad. And these were actually super helpful for me because I also uh, basically benefited from this, and the reason I'm in research now is because of these scholarships. So there is one called MyTax Global Link Summer Research Scholarship Program. This is a collaboration between Canada and India, where you're fully funded to go and do research for 3 months in a Canadian university. And I was a beneficiary of this. Um, and the second is to do research in USA in University of Southern California. It's called the Viterbi Indo-US Science and Technology Forum Program. This is again to do research internship, summer research internship after your third year or beyond, I think, like after third or fourth, I think. And this is also a very good opportunity. I know some of my seniors went and did there, and yeah. The third one is in Germany. It's called DAD Scholarship, and this is usually for people who have very, very, very good GPA. So if you're one of the toppers, definitely check it out. That's just one aspect. I mean, like, I'm not discouraging people who don't have good GPA to apply to it, but yeah, that was just my experience when I was in undergrad. So this is to do research in Germany, and this scholarship is available to do summer internship in your undergrad, or to do your masters after undergrad. So it's for both. And the next one is called Charpak scholarships. That's an MOU that India and France have, like an understanding that the two countries have. And they also again offer scholarships to do research in summer as an undergrad, or to do pursue your masters. So yeah, these are just some helpful info that I was told as an undergrad, which benefited me. So I thought I'll share it with you guys. Um, and acknowledgements, I would like to really thank, first of all, my professors, Professor Ione Fine and Professor Jeffrey Boynton, who have guided me throughout this entire research program. NEI and NIH, which is the main source of funding for our research. And Professor Michael Baylor, Ariel Rockham, all of these people have been super helpful for, like, random questions about modeling and so on. SG and Nil. SG was my teammate. Nil is my undergrad who helps me with data collection and all of the vision and cognition lab at UW. And I would like to express my sincerest thanks to ACS College of Engineering staff for giving me this opportunity to address you and share some things that I learned over the course of my PhD. So yeah, that's my talk, and I can take questions now. I don't know how you want to do it. Like, do you want to maybe give your question, like ask the question to your professor, and they can relate to me? Um, uh, okay, I'm done. Thank you. I think all of you are muted. I don't know if you're talking. Hi, what's your name?
Hi, my name is Mohammed Ma'am, and I am the
Okay,
speaker.
nice to meet you. Mm,
On behalf of our principal sir and our staff and everyone present here, I extend our heartfelt thanks to the speaker for making the session so informative and engaging webinar, ma'am. So your
thank you.
insights on the topic have expanded our understanding in significant ways, ma'am. So I would like to thank from everyone behalf, and yeah, we are looking forward for the knowledge from you in the future also. So thanks from everyone, and that's all.
Of course, thank you so much. Oh, if you have any questions about the talk or anything in general, feel free to come up and like ask the question I have. I think, do we have time? I think, do we have another 15 minutes, or are we done over time?
Yeah. That-
Actually, I have one question.
Oh, sure.
Can
Yes.
so we have this retinal pigment result. So we are
Mhm.
working on glaucoma. Like
Okay.
in glaucoma condition, we are using the fundus images, and
Mhm.
on the, like we are working on changes in cup disc size, optical nerve changes like that. So can we work on this retinal pigmentosa using the fundus images? Can we take up that as a project?
Uh, I think it depends on the problem statement. You can use fundus images if you want to characterize the changes in the retinal anatomy itself, and that is definitely one of the very useful aspects which clinicians really want to know, right? Because a lot of times. In a diseased retina, it can expand or contract, it can change the underlying circuitry, and all of these things will lead to perceptual distortions and so on. So it is definitely useful from a clinician's perspective, where they want to understand how the anatomy changes in the retina. However, and that can be like the first step to understanding what individual changes you need to make to these site recovery technologies to elicit cell responsiveness and functional vision in people. And having said that, glaucoma is a condition which affects the optic nerve. Right, so you won't be able to use retinal implants or optogenetics in the retina to treat that condition, because the damage is beyond the retina. So if whatever you change in the retina, it won't make a difference because optic nerve is cut off. So actually, one of amongst the available sight recovery therapies that are there now, cortical implants may be your best bet right now, because there are like Neuralink, Elon Musk's Neuralink, for example, is working on that, and there are actually many other companies which are taking a more scientific approach to this, where they are trying to use the same electrical implants, but instead of the retina, to the brain directly. And I think that is a therapeutic result that you can have for glaucoma, because you're treating it beyond the damage area. But the fundus images that you have, that will definitely give an idea to a clinician as to how the retinal anatomy changes due to this damage. And there are a lot of other additional problems where when something goes wrong or in glaucoma, that will lead to secondary issues like infection inside the eyeball, like in the aqueous humor, like the cells start degenerating even in the retina because it's not getting used, or the environment around it has become toxic, and so on. So definitely understanding fundus images or ERG images would be a good way to understand how the anatomy changes, as far as I understand.
Thank you. Thank you so much. Any questions? Any more questions?
Hello.
Hello.
Vaishnavi, thanks for accepting our invitation and sharing your valuable thoughts and research things what you have. Okay, struggle to achieve something. So easily you have shared that knowledge to our students. And on behalf of my management and head of the department of biomedical engineering and faculties, okay, and students, we are very much thankful to you and for your patience while presenting and everything starting from how the eye okay is there, or it is the construction, or by nature what is eye, and other things you explained very nicely, and your research related things also you have presented very well. And I hope our students are going to make use of your efforts, okay, whatever the the different funding agencies are there encouraging for the research and other things now, that also nicely are presented. Only now the rest of the things is left to our students, how best they are able to catch hold of those details what you have given. And uh, we are very much thankful once again. Okay, thank you, Vaishnavi, thank you.
Definitely. Thank you so much for giving me the opportunity to address your students and share my work. Yeah. Thank you very much for sitting patiently listening to the whole thing, and mainly to ACS College of Engineering staff for making this possible. So thank you.
Thank you.
Okay.
Thank you.
Bye.
Thank you.